Thank the LACNet's committee and panel for introducing me and inviting me again to talk about this briefly but increasingly relevant topic, genomics in neuroendocrine tumors. So what is genomics? It all starts with the gene, and I apologize if this is reviewing a lot of old material that you may know, but it's essentially a segment of DNA, and it serves as the template that allows us to build the proteins and molecules that allow us to do what we do. And the genes are condensed into structures called chromosomes, as you can see in the diagram there. They're contained in the nucleus of cells, and cells, as you guys know, are the building blocks of the tissues and organs that make us who we are. So, you guys may know of the Human Genome Sequencing Project, and this was the first attempt, a global and took almost a decade effort to map out the entire collection of genes in the human. And the first draft was actually finished in 2001. It took about $2.7 billion, global effort, but at the end of all this, we had our first human reference genome of approximately 3 billion DNA bases. Now, why is it important to know what the human genome is? Why do we need to map it out? Because in order to know what's abnormal, you first must know what's normal. And this is essentially what genomics is. It's the study, the mapping of all the collection of an individual's genes, equal the genome. So, since the time of the Human Genome Project, the actual machines, the technology used to map out the genome is actually a little bit more advanced. We've developed some new technology, and these are called the so-called next generation sequencers, because they are the next generation coming after the original sequencers used for the Human Genome Project. You may be hearing terms like massive parallel sequencing, high throughput, molecular profiling. These are all terms to express that we have simply been able to now map out genes and sequence genes at a much faster, highly efficient, massive rate. And the actual first of these next generation sequencers, as I've highlighted here, was the Roche 454. With the reference genome in hand and with these next generation sequencers, by 2006, you were able to completely sequence an individual's personal genome for about $14 to $25 million. Now, right around this time, a group of privately funded and some government-funded groups sought out to now systematically catalog all the mutations in a wide variety of adult and pediatric cancers. So now we're moving from all the genes, the personal genome, to now the cancer genome. These are the groups that I've highlighted here. And now in the current decade, technology has become increasingly commercialized. Molecular profiling is now available by a lot of different companies using different platforms. I've highlighted here some, but not all, of the commonly seen companies that you may see performing next-generation sequencing. I've also highlighted here some of the sample requirements. And I didn't mention this at the beginning, but NGS, or next-generation sequencing, or molecular profiling right now is actually done majority of the time on tumor tissues. These are tissues of your tumor obtained either from surgery or biopsies that are stored in this preservative called FFPE, which is the most common of the preservatives that the samples are being run for molecular profiling. I've highlighted the type of sequencers or the different names of the machines that they're actually using for this sequencing. And then companies differ based on the number of genes that are covered. Some companies express that they covered 315 genes, some over 500 gene mutations. Costs, as you can see here, we've now moved a little bit better from the $1 million genome to about a couple of $1,000s because of the fact that we're no longer sequencing a whole array of genes, but rather those associated with cancer. And also important to the patient is how long does NGS or molecular profiling take. And here, as you can see here, usually on the order of 1 to 3 weeks is the most common across all these companies. And I just wanted to highlight that most of these NGS companies that I'm talking about are actually testing for somatic mutations. These are mutations in your genes that develop throughout your course, your life course of disease, rather than those that are hereditary or passed down from the germline. And there are separate tests that are testing germline mutations. So I wanted to tease that out. This is two different type of concepts that are important to know. So I've given you a timeline and a little history of genomics, but why should we do genomics? Why should we send our tumor tissues for next generation sequencing? Well, conceptually, it really boils down to 3 reasons why. The most important reason, most obvious reason, is for clinical decision making. The other 2, to identify disease-causing genomic alterations, is pretty self-explanatory. And then I'm going to go into a little bit of how important genomics is for research purposes. But for clinical decision making, how can it impact us right now in the clinic right now? There are currently a couple of FDA-approved therapies here that I've mentioned, and these are granted rare, but they do require some molecular profiling to detect these. And this is microsatellite instability, which predicts benefit to immunotherapy, and NTRK gene fusions, which definitely require NGS. These types of mutations do not comprise the majority, but when they do, in patients that have been treated with all these standard therapies, offer additional options and are FDA approved. But more commonly, mutations that we detect in genomic profiling can help us enroll in clinical trials. There are 2 trials right now, MATCH or TAPER. These are both accruing here at Cedars as well. What we're doing is trying to match available targeted therapies to specific alterations detected in your tumor. And I've listed a host. I won't go through a lot of the alterations that we're looking for in this type of trial. So what about research purposes? I want to give you a kind of a flavor of what we're doing with the NGS and next generation sequencing in neuroendocrine tumors, particularly PanNets. Here we can see we profiled the tumors of 46 patients. You can see the majority were stage 4 or metastatic disease. Majority were lower intermediate grade. The most common site of spread was to the liver. Now, here's what we found in terms of the most common alterations we found in our cohort of PanNets. We found that 2 mutations in MEN1 and DAX, and the middle category there tells you what their role is in the normal body. These are chromatin remodeling involved in remodeling the structure of the chromosomes. These are the most common, but as you can see here, there were other alterations that were commonly found. Specifically, I want to point out to the couple of mutations involved in mTOR signaling. As you can see here, these mutations were pretty prevalent, and it also supports the fact that why maybe drugs that target mTOR, as you may well know, everolimus is one such, might have proven efficacy in PanNets. And then we also identified some proteins beyond genes that were involved in mTOR signaling, some in immunotherapy, but I wanted to highlight a couple of them, interestingly, were associated with resistance to chemotherapy. Now, what's the point of finding frequency of genes? What are we going to do? How are we going to correlate to outcome? So our next part was what we found was that there were some genes, particularly RB1 and TP53, were more associated with a high-grade tumor. There were those, conversely, MEN1, which were more associated with non-metastatic disease or low-grade tumor. And when we actually follow these patients out, you can see that there are patients, those in the red, that had the presence of mutations in what we call the replicative stress pathway. These are proteins that are involved in making sure that DNA replication occurs normally, mostly TP53 and RB1, as I highlighted before. As you can see, those in the red bar, those that have these mutations, those patients tended not to live so long compared to those in the blue bar, which tended to live a little longer. So now I wanted to also reassure everyone that it wasn't only in PanNets, but NGS studies are now kind of exploding into other arenas. Pulmonary carcinoid tumors, there are NGS studies in small intestinal neuroendocrine tumors. There are those in patients with a variety of other neuroendocrine tumors as well. And then another concept I wanted to end with was that these were all in tumor tissues. You might hear of the concept of a liquid biopsy. And what this is it's no longer profiling genes based on tumor tissue, but actually in the blood of patients, because you often have circulating tumor DNA that could be detected. And it's been established in other tumor types. In neuroendocrine tumors, this was one of the earlier studies, and what did it show? As I've highlighted here, All the higher amounts of circulating tumor DNA was detected in patients with metastatic disease. As you can see to the left, those with non-metastatic disease tended to have less amounts of circulating tumor DNA. And this was an early indicator that circulating tumor DNA could be predictive of tumor volume, how much tumor you had if your tumor was growing. As you can see here, those with metastatic disease spreads to other sites have higher levels of circulating tumor DNAs. And this is one of the first steps of developing circulating tumor DNAs in this arena. And lastly, I'd like to end with the future of genomics. What's it going to look like? Well, these are exciting developments, but I do believe that these are still small subsets, as you can see, small cohorts of patients. We need to validate them in larger samples of neuroendocrine tumor patients to truly define a genomic mutation that is both prognostic and or predictive of targeted therapies that are available. And ideally, clinical trials should be a consideration for matching targeted therapy to genomic profiling. And I like to list that there is not just one or two, but there are many ongoing clinical trials throughout the nation seeking to identify novel targets for additional options of therapy. Thank you.